continue our discussion on amino acid properties, and in this particular class we are going to understand the spectroscopic characters. In the ones that we considered previously, we looked at amino acid properties in general in terms of the side chains, in terms of the presence of the heteroatoms, whether we had only carbon and hydrogen for the side chains or whether we had oxygen and nitrogen or sulfur in them. So, we need to know which amino acids are ionizable, which amino acids have basic properties or acidic properties, and in the last class we looked at the isoelectric point in amino acid titrations, and we tried to understand where this neutral part of an amino acid comes from in terms of what we know as the zwitterionic form. We also looked at the hydrophobicity in terms of a hydropathy index. We were looking at a specific way to demonstrate how we can find out a hydropathic or hydrophobic region of a protein. So, these are interesting properties of amino acids, and they go in a very long way to understand how proteins behave, because these are the building blocks of proteins. In this lecture, we are going to look at the spectroscopic properties of amino acids. Now, what do we mean by these spectroscopic properties of amino acids, and what are the amino acids that are going to contribute to the spectroscopic characteristics? So, when we look at the different keywords that we have looked at so far, we have not seen what chromophores are yet. So, what are these chromophores? A chromophore is a chemical group that absorbs light at a specific frequency, and what it does, it imparts color to the molecule. Now, or we can also define it as a specific group of atoms in a molecule that comprise the orbitals involved in the transition that constitute a chromophore. So, what do we mean? In general terms, if we have two energy levels, we are going to have a transition. This transition is going to correspond to a specific energy. This energy we know is related to the wavelength by the following expression. So, when we look at this expression, we look at the wavelength, we look at the energy, and we try and figure out that if there were particular chromophores in proteins, what would the lambda values be and where would they lie in the overall electromagnetic spectrum. Now, what do we mean by this? When we look at the electromagnetic spectrum, we see that it has a range of wavelengths. The range of wavelength means it has a range of energy. Now, this means that when we look at the specific wavelength values, we know that we have the visible region from 400 nanometer to 700 nanometer, and we know the colors that we follow here, the VIBGYOR that we know. Now, we have the ultraviolet region that corresponds to a specific wavelength that is going to be less than 400. We know that we have the IR region that is going to be the region beyond 700 nanometers. This means that when we look at the specific bonding or the specific electronic availability or the transition of these electrons, we have to figure out where these transitions are going to lie in terms of our electromagnetic spectrum. So, what are these energy levels? If we look at a generic form of these energy levels, we have the sigma orbital, we have the pi, we have the non-bonding, we have the pi star, and we have the sigma star in terms of the way they in terms of increasing energy. Now, based on a transition, what do we have in the first transition here? Sigma to sigma star. That means that if this is our ground state where we have all our electrons sitting in the sigma, pi and non-bonding, what is going to happen when we have a sigma to sigma star transition? It means that the energy level is, this is our energy jump. Similarly, when we go from n to sigma star, so these are the electrons we are talking about, the transitions that we are going to talk. We are going from n to sigma star, that means we are losing one from this particular point and we are going up to sigma star. Here we are going from pi to pi star, so this is the value. Here we are going from n to pi star, which means this is the value. Now, we realize that each of these transitions involves a different energy value, which means they belong to a different wavelength value, and this wavelength is important into understanding where in the electromagnetic spectrum do we fall. Is it in the visible region? Is it in the IR region? Is it in the UV region? And then how can I measure this? So, if we look at the transitions, we see that the n to pi star is less than the pi to pi star, less than the n to sigma star, less than the sigma to sigma star in terms of the value or in the terms of the energy levels that we are talking about. Now, if we look at the energy levels that we saw that the sigma to sigma star transition involves very high energy and usually lies in the vacuum UV region. Now, how was that known? Because we are from the energy value, we can calculate the lambda, and from the lambda value we can determine which region of the electromagnetic spectrum this falls into. So, we have a saturated hydrocarbons that undergo sigma to sigma star transitions only and show absorption bands in the 150 nanometer wavelength. Similarly, when we look at compounds that have an unsaturation, what do we mean? They have double bonds in them or even a lone pair of electrons. They undergo pi to pi star or n to pi star transitions. They absorb at higher wavelengths that are there in the far or near UV regions. So, what does this mean? This means that the value of the transition that we are looking from pi to pi star or from n to pi star or from pi to pi star, the energy value that we are looking at here has, so this is a particular energy value and we have the lambda value. The lambda value is in the far or near UV region. This region is important in all our biochemical observations and we will see why. Because we have a group of atoms in a molecule, we know this chromophore is going to be what is important to us in understanding what the chromophores are in proteins. So, when we look at the chromophores in proteins, these are the aromatic residues. Which are the aromatic side chains? They are the phenylalanine, the tyrosine and the tryptophan. So, just like we had looked at all the representations of the amino acids, we have what is called, we know that this atom is called the C alpha carbon. We have the NH2 attached here and the COOH here. This is just a representation. We know that when we look at this form, if it is in the zwitterionic form, this is going to be NH3 plus and CO minus. But just for a common representation, we have put it as NH2 and COOH. Then here we have tyrosine, which has an OH group attached to it, and here we have tryptophan, which has an indole group. So, these are the aromatic residues that are present in proteins. These aromatic residues are the chromophoric moieties in proteins that bring about the spectroscopic characteristics to a large extent in proteins, making them useful for an understanding of their concentration, their presence and so on and so forth. So, we look at the near UV region. What do we mean by the near UV region? We know that the visible region starts from 400 nanometer to 700 nanometer. So, the near UV region is close to 320 nanometer. So, the chromophoric part in tryptophan, tyrosine and phenylalanine is responsible for the near UV region with a maximum absorption around 280 nanometer, which corresponds to the pi to pi star transition. So, this is what is important in an understanding that we have the 3 aromatic amino acid residues that are going to contribute as chromophores in proteins, and they are going to give a maximum absorption around pi to for the pi to pi star transition that is going to fall in the region around 280 nanometer, which is the near UV region. The disulfide bonds that we know of, what are these disulfide bonds? We will learn in a later class, but we know that the cysteine residues can form what is called cysteine, which I will show you in a moment, and they have an absorption around 260 nanometer, and they also can serve as a potential chromophore, because again we see that 260 nanometers also falls in the region of our near UV as we saw. So, when we are looking at the near UV region, we have this from 250 to 400. We know that from 400 we start the visible region. So, close to the visible region we have the near UV region, followed by the far UV region from 190 to 250 nanometer. This is also important in biochemical techniques in terms of characterization of proteins, and we have the vacuum region, that vacuum UV region, which is less than 190 nanometer. So, the wavelength range that we see is going to be dependent on the energy levels. What are the energy levels dependent on? They are going to be dependent on the specific transition that we are talking about. So, whether we talk of an N to pi star transition or a pi to pi star transition, this difference in energy that we have, the difference in the energy levels corresponds to a particular wavelength, and if this wavelength falls within these regions, we will be able to observe these transitions in a UV spectrophotometer. Now, in this understanding, this is where we were talking about the cysteine. So, this is the amino acid cysteine, where we have this as the SH. What is the SH? That is the CH2, SH is the side chain. Okay. So, we have the alpha carbon. So, let us try and identify. So, here is our alpha carbon, here is the hydrogen associated with the alpha carbon, here is the amino part, and here is our acid part. So, this is the C alpha, this becomes our C beta, this becomes our sulfur, and here is attached a hydrogen. Now, what happens when we have the loss of the hydrogen in the formation of the disulfide linkage? We have a particular moiety that is also going to absorb, as we saw in the previous slide, around 260 nanometers, also contributing to being a potential chromophoric moiety. Now, if you look at the absorption spectra of the amino acid residues, now what do we mean by this absorption spectra? If we take the amino acid tryptophan or tyrosine or phenylalanine independently in what is called a cuvette for the machine known as the UV spectrophotometer, and we scan a particular wavelength. So, we are scanning from 200 to 400 nanometers. So, when we scan from 200 to 400 nanometers, which region are we scanning? We are scanning around the near UV region. Why do we want to do that? We want to see whether we get any specific absorption due to the scanning. What do we see? Yes, we do have absorption associated with the scanning from 200 to 400 nanometer, and we see an absorption around this region. So, we see, what do we mean by the absorption? We mean that we have peaks, and these peaks tell us that yes, tryptophan is present, tyrosine is present, phenylalanine is present, and this gives us an idea of where we are to look at to see whether we have a protein present, because if the protein solution or the protein of interest has any of these aromatic amino acid residues present in them, which is most likely true, then we will see a presence of a peak around the region of 280 nanometers, telling us that this is a protein having this amino. So, there is our lambda max. Where is it? It is around 280 nanometers, telling us that our solution does have a protein or does have an amino acid present in it that is going to correspond to 280 nanometer as a lambda max, telling us the presence of a protein. Here also we see some transitions following at a lower wavelength, meaning a higher energy, meaning a different transition, but we are interested in this particular transition of 280 nanometer, because the absorbance value of this particular wavelength is used to measure the protein concentration, which we will see in a moment. So, when we look at the far UV, this is usually used for proteins and peptides that do not have aromatic residues as chromophores, because when we are talking about a chromophoric unit, we are saying that the aromatic residues, the presence of the aromatic amino acid residues, that is the tryptophan, the tyrosine, the phenylalanine, these are going to give us specific transitions at 280 nanometers that make it useful for us to identify whether we have a protein solution or whether we have these particular peptide or not. But what happens if our particular peptide does not have any of these aromatic amino acid residues? Then we have to resort to looking at the far UV region to identify whether we have a particular peptide bond. Now, the peptide bond is again a major chromophore in the far UV region. Why? Because in this particular case, we are also going to get what is called a pi to pi transition and an n to pi star transition. The strong absorption band is around 190 nanometer, that is for the pi to pi star transition, and there is a weak band around 220 nanometer for the n to pi star transition. Now, these transitions occur for the peptide bonds. All of the protein, when we are going to study the interactions or when we are going to study the formation of the peptide bond in the next lecture, we will see that this peptide bond is present throughout. So, the identification of the peptide bond is going to mean that with a lot of these peptide bonds that are present, we are going to have a large absorption band at 190 nanometer and at 220 nanometer. So, for the identification of proteins, we usually resort to the aromatic amino acid residues, where we are looking at tryptophan, tyrosine or phenylalanine, because these are relatively less abundant in proteins than the peptide linkage, which is present definitely in proteins because they link the amino acids together. Now, a problem here is that oxygen also strongly absorbs at 190 nanometer. So, we measure usually the absorption at 205 nanometer, where we have the molar absorption coefficient of the peptide is actually half that observed at 190 nanometer, which is actually the strong absorption band. So, ideally when we are looking at a peptide that does not have an aromatic amino acid present, then it is preferable to measure the absorbance at 205 nanometer rather than 190, despite the fact that the strong absorption band occurs at 190 nanometer. Why? Because oxygen also strongly absorbs at this point and it will interfere with our measurement. So, if we look at the absorbance of the aromatic amino acids, we look at what is called an extinction coefficient that gives us a measure of the capability of the particular moiety or the particular chromophore in this case to absorb light. Now, when we look at the extinction values here, we know that these are the side chains of tryptophan, we have tyrosine and we have phenylalanine, and this extinction value is given in the units of liter per mole inverse per centimeter inverse. Now, what is this, or what are the lambda max values? The lambda max values are the particular spectrum of the amino acid listed here, and we look at the lambda max value corresponding to where the peak is observed in the particular spectra that is taken for the amino acids. Now, when we look at the theoretical estimation of this epsilon value, then we are looking at a very important aspect in trying to understand how this spectroscopic characteristic is used in biochemistry for what? For the observation and the measurement of the absorption of oligonucleotides, peptides and protein solutions, all in the UV range. Why do they fall in the UV range? Because all the transitions that we observe from the N to pi star and the pi to pi star, that are the most common ones that we observe, the energy transitions or the difference in the energy levels falls in the UV range for the particular wavelength that we observe. We look at the Beer-Lambert's law, which is something that you have probably studied already, where we have the absorbance value, and from the absorbance value we can, from a measure, determine the molar extinction coefficient of a specific solution, given that we know the concentration of the protein that has been taken and the path length, which is usually 1 centimeter for the cuvette that is used in the machine. So, this is important when we are trying to understand a theoretical estimation to figure out what the extinction coefficient of a particular protein is going to be at 280 nanometer, given that we know the number of tryptophan, that is W, we know the number of tyrosine, that is Y. So, we have W and Y. W is the 1 letter code for tryptophan, Y is the 1 letter code for tyrosine. Now, given the value that we had for the extinction coefficient for the specific amino acids, these are additive values. So, if we have an NW, meaning the number of tryptophan residues that are present in the protein, NY number of tyrosine residues in the protein, we can get an estimate of what the value of this extinction coefficient is going to be, given that we know the number of tryptophan and the number of tyrosine. Now, given that we know or we can determine what the absorbance value is from the machine when we find out the OD of the optical density value from the machine in the and then given the extinction coefficient that we are calculating from a knowledge of the number of tyrosine and tryptophan, what we can do, you realize, is we can find out the concentration of the protein, a very, very important factor, a very, very important calculation, a very common calculation that is done in any protein chemistry laboratory, because you have to determine the protein concentration in any experimental observation that you are going to make. So, let us look at a simple problem based on this. So, the problem states that we have to calculate the molar extinction coefficient for a 50 amino acid peptide. Now, what is this amino acid peptide? Now, we have looked at a 50 amino acid peptide that contains 3 tyrosines and 2 tryptophans, and it has a molecular weight of 5.5 kilodalton. So, there are certain new terms that we are learning here. One is the kilodalton. Now, what is the kilodalton? The kilodalton tells you a dalton is 1 unit of molecular weight, 5.5 means 5,500 kilodaltons. So, when the question is that we have to calculate the extinction coefficient and the absorbance for a 0.1 millimolar solution of the protein. So, the first thing to try and find out what the extinction coefficient is, means we need to know N, we need to know what NW is, what is NW, and we need to know what is NY. So, NW we know is equal to 2 because there are 2 tryptophans, and NY is equal to 3 because there are 3 tyrosines. So, the molar extinction coefficient for tryptophan is this, for tyrosine is this value. Then according to the formula that we looked at previously, we have the value of NW here, we have the value of NY here. So, we can find out the extinction coefficient for this particular set or this particular peptide. So, the number of corresponding residues is 2, as we learned, 2 tryptophan, 3 tyrosine, and there is our value. So, once we have the molar extinction coefficient, we know the concentration, we know the path length, we have to take the concentration, it was given as 0.1 millimolar. So, we have to be very careful that we, because our units have to cancel out, absorbance values are unitless. So, we have an absorbance value 15220. Where did we get this from? We got this from an understanding of the tryptophan and the tyrosine, and the concentration, the path length giving us an absorbance value. So, that is the absorbance value. So, the peptide containing 50 amino acid residues has a specific extinction coefficient corresponding to 15,220 mole inverse centimeter inverse and an absorbance value of 1.522. This is used for the determination of the concentration of proteins, as we mentioned, and we can calculate the exact concentration of many proteins given we know the extinction coefficient. So, the absorption spectra depends upon the protein concentration, solvent buffer, the temperature, which we will see as we go along in an understanding about how we can apply the UV spectroscopic studies to proteins in general. Another small thing that needs to be covered, or just an understanding, that there are some modified amino acids that are just, as you can see the names, hydroxyproline, carboxyglutamate, phosphoserine and phosphotyrosine. And there are other combinations that are called post translation modifications that occur after a protein is synthesized, where we have a combination of amino acids that sometimes give rise to a different moiety altogether. So, the amino acid properties that we studied were the isoelectric point, the amino acid titration, the hydrophobicity and spectroscopic properties of the amino acids that give us an understanding of amino acids in general. Our next lecture will be on the combination of amino acids now in the formation of a peptide bond before we embark on an understanding of protein structure. Thank you.